Hello, I'm by the names of Rosina Nibikora. I'm a student of INES in Biomedical Laboratory Science, Faculty of Health. I'm gonna present about assignment of applied immunology, where my task is from 31 question to 35 question. First question is asking about principle behind agglutination reaction. Second one is asking about how latex agglutination work in diagnostic. Third one is asking about principle of precipitation reaction in immunology. Fourth one is asking about how does radioimmunodiffusion test performed. Fifth one is asking about principle of ELISA technique. Let me start by question 1, which is asking about principle behind agglutination. First of all, agglutination reaction is technique based on a specific interaction between antigen and antibody to make a complex and result in formation of visible clump or aggregates. This agglutination depends on three principal reaction, where one is specificity. In specificity is where antibody recognize and bind to unique epitope of antigen, meaning every antibody bind on a specific epitope of antigen. Other one is cross-linking, where antibody it bind to antigen to form a cross-link between particles. Third one is lattice formation. This lattice formation is where multivariant antigen bind or interact with multivariant antibody to create stable lattice structure. Here I have provided types of agglutination, which are direct agglutination, reverse or passive agglutination, and the hemagglutination. In direct agglutination is where antibody react direct with antigen on the surface of the cell to form aggregate or to form that cramp. This can be done in ABO and lysis grouping. Second one is reverse agglutination, where antibody attack to innate carrier, then that antibody coated particle agglutinate when encounter the target soluble antigen. Hemagglutination is agglutination where we use red blood cells as carrier of antigen to make a reaction between antigen and the RBCs or red blood cells. Here I have image show that those types of agglutination and like procedures. Here indirect is where we take antibody and bind it to antigen directly to make the cramp. In indirect is where we use antihuman globulin. We take antibody and bind it to antigen and there is no agglutination. For agglutination, maybe that antibody is hidden, like IgG, maybe is hidden. So to make that to confirm if that antibody is not there or is there, we add antihuman globulin or AHG to see if we can find agglutination. That process is called indirect antiglobulin test. And here there are some applications of agglutination. That agglutination is used in blood grouping like ABO and lysis grouping, is used in infectious disease diagnosed like for detection of bacterial and viral infection such as streptococcal infection, and is used in pregnant test like to detect human chorionic gonadotropin, which is a hormone produced during pregnancy. and is applied in autoimmune disease diagnosis, where we identify autoantibodies against self antigen in diagnosis of conditions like rheumatoid arthritis and systemic lupus erythematosus. And this agglutination is used in virology, where hemagglutination assay help to detect virus that bind to red blood cells such as influenza and rubella virus. Let me go to second question, which is asking about how does latex agglutination work in diagnostic. First of all, latex agglutination is a diagnostic technique that reveals the specific interaction between antigen and antibody to detect the presence of disease markers, that disease such as pathogen or immune response in patient sample. How do we perform that latex agglutination? First of all, there's preparation of latex bed, where latex bed are coated with antibody to detect the antigen or antigen to detect antibody. Means we coat that board according what we want to detect. If we want to detect antibody, we will coat that board with the antigen. If we want to detect antigen, we will coat that board with the antibody. Secondly, we mix with patient sample, which can be blood or urine or cerebrospinal fluid or other body fluids. Thirdly, there is formation of antigen antibody complex, where sample contain target molecules. It will bind to the coated latex bed to form cross-linked network of bed and the analytes. There is visual detection, where agglutination is visible to our naked eye or under microscope. Means when we are visualizing this visible, we visualize the agglutination which is visible by our naked eye or which is visible when we are looking under microscope. The positive result will indicate formation of cramps in the suspension, while negative result will show smooth or uniform solution without visible cramps, means we not being agglutination. This latex agglutination in a diagnostic, it has like three types. There is direct latex agglutination, indirect or passive antibody in latex agglutination, and the reverse passive latex agglutination. Indirect is where used to detect antigen in the patient sample direct. In indirect, it is used to design or to detect designed to detect the samples. Here in reverse passive latex agglutination, latex bed are coated with antibody to detect soluble antigen such as toxin or protein, and is common in clinic. Use this to detect CRP or which indicate inflammation or which is inflammatory marker. Here there is like some procedures on picture. Those procedures I explained above, they are summarized in that picture, where glycoprotein 43 is added to latex to form glycoprotein 34 CRPS. When we add circulating antibodies to form agglutinated complex between that antigen glycoprotein 33 and the antibody we added there. On we start by we code that bed with antigen to detect antibody, where we take antibody and add latex particle to form complex and add that glycoprotein which is circulating antibody to form agglutinated complex. There is application of that latex agglutination in a diagnostic. First of all, is applied in infectious disease detection. means that data's agglutination can quick identify bacterial or viral infection. For example, it's used to detect group B streptococcus in pregnant women or meningococcal antigen in cerebrospinal fluid. Also, it is applied in autoimmune disease diagnosis, like in rheumatoid arthritis diagnosis, and also is used in inflammatory marker, like to detect markers like C-reactive protein, which indicate inflammation or infection. And that latex agglutination can be applied in pregnancy test for detecting presence of human chorionic gonadotropin in the urine as indicator of pregnancy. Allow me to go on that question, which is asking about principle behind the precipitation reaction in immunology. First of all, precipitation reaction is specific interaction between a soluble antigen. Here, what I did mention that agglutination it use solid antigen, while this precipitation use soluble antigens. So is a specific interaction between the soluble antigens and the soluble antibody to form a soluble antigen antibody complex. This complex aggregate into large structure that precipitate out of solution and create visible or measurable precipitate in solution. Mechanism of precipitation reaction. First of all, it is antigen antibody binding, where soluble antigen and antibody are mixed. Then they bind specifically to form antigen antibody complex, and both antigen and antibody must be multivariant, having multiple bind site or to allow cross link. Second mechanism is lattice formation, where precipitation occur. Antigen and antibody must be in optimal ratio, means must be in balance, where the amount of antibody have to be equal to amount of antigen to create a stable lattice network. Here we have like post zone, plus zone, and zone of equivalence. Here that lattice formation will be formed in zone of equivalence where we have equal amounts. So this network grow largely and eventually become insoluble and forming visible precipitation where the optimum ratio is found in zone of equivalence like I was talking about, where antigen and antibody concentration are balanced. Here there is mechanism of how it is performed, where we take antigen and we add antibody in a solution where antigen are soluble and making antigen antibody complex to observe agglutination or formation of precipitate. Here I have provided like the two types of precipitation reaction, where it is done in solution or link test and done in imine electrophoresis. In solution is where we have gel-based precipitation or immunodiffusion. In this technique, antigen diffuse into gel contain uniform distributed antibodies. In immunoelectrophoresis is where combine electrophoresis and immunodiffusion for detecting abnormal protein in disorder like multiple myeloma, which is cancer. Application in diagnostic is applied for detection of specific antigen or antibody and is used for quantification of immunoglobulins such as IgG, IgA, and IgM in patient serum. Also, precipitation reaction is applied in identification of protein As we have seen, it is used in immunoelectrophoresis, and electrophoresis can help us to count or to identify protein by help identifying abnormal protein for diagnosing condition like multiple myeloma or autoimmune due to autoreactive. Fourth question is asking about how does radioimmunodiffusion test is performed. First of all, radioimmunodiffusion is a quantitative immunological technique used to measure concentration of specific antigen in a sample. This radioimmunodiffusion it helps us to quantify or to know amount of antigen which are present in the patient sample. This radioimmunodiffusion based on precipitation reaction between a soluble antigen and antibody. When an antigen diffuses radially from a well into a gel containing a specific antibody, it forms a precipitant link where antigen and antibody reach to zone of equivalence where there is optimum antigen antibody, means balance between antigen and antibody, their amount are the same. And the diameter of this link is directly proportional to the antigen concentration in the sample. Here there is how it is performed, where we have antigen bind it to antibody which are soluble and make like precipitant because it based on precipitation. Here we have left side and right side. We have antigen, we have a when coated, and we have antibody and the slide where that process is done, and that agar as a media of where antibody and antigen are going to interact. And from left to right side, concentration is decreased, means concentration of antibody antigen is decreasing. There is procedures of how we perform precipitation reaction. No, radioimmunodiffusion. Procedures one is preparation of gel plate. Here, agarose gel prepared with specific antibody and put into petri dish on a glass plate and allowed to solidify. Also small circular wells are carefully punched into the solidified gel. The second one is loading samples, means known concentration of target antigen are added to separate well and an unknown sample of patient or test sample are added to remain well. Incubation. Here that it will be incubated at optimum temperature which is 25 to 37 for 18 to 48 hours, where antigen diffuse radially from well to into the gel and interact with that antibody coated in the gel or coated in the well. Fourth one is formation of precipitation link. As antigen diffuse outward, it bind with antibody in the gel to form insoluble antigen antibody complex. Means precipitant is going to be formed from antigen antibody complex. Fifth one is measurement and quantification. As we know that radioimmunodiffusion is a quantitative method used to quantify antigen in the sample. After incubation, the diameter of each precipitation link is measured using ruler or caliper. And that radioimmunodiffusion is applied in quantification of serum protein, where it helps us to diagnose and monitor conditions like multiple myeloma. Second one is it used to detect the complement protein, where that immunodiffusion quantify complement component such as C3, complement 3, complement 4, which are important in the diagnosis of immune diseases like systemic lupus erythematosus. Third one is monitoring immune disorders, where it is used to assess patients with suspected immune disorders. Fourth one is quality control in biopharmaceutical. This immunodiffusion can be used for measuring concentration of specific protein in a vaccine and in antibody production. Let me go to last question, which is asking about what is principle behind ELISA technique or principle of ELISA technique. First of all, this ELISA is enzyme liquid immunosorbent assay, which is used for immunological method to detect and quantify specific antigen or antibodies in biological sample. This ELISA, principle behind ELISA, this ELISA uses specific bind between antigen and antibody to detect a presence of target molecule. This ELISA use enzyme conjugate antibody or antigen which upon reaction with a specific substrate and proceed a color change or a damageable signals. The intensity of this signal is proportional to concentration of target molecule in the sample, which allow both quantitative and qualitative analysis. The procedures of ELISA, how ELISA is performed. First of all, we take a sample on a well, means we add a sample on a well. We add there a conjugate antibody, means antibody with an enzyme. Third, we incubate and wash. We incubate that conjugated antibody with sample, then we wash. After wash, we add a substrate which provide the color. Example of that substrate is chromogenic or chromogen. For and incubated in 10 minutes, that substrate is therefore giving color. When color change, we interpret result, and when there is no color, we have interpretation on that result. Fifth, we add the stop solution. This stop solution is there to stop reaction between that substrate and enzyme which is combined with antibody, because when there is prolonged reaction between substrate and enzyme, it can affect the results. Sixth is reading result, how the reaction is looking like, if there is color change or if there is no color change, or quantify the amount of antigens present. Here I have different types of ELISA, like 4 types. First one is direct ELISA, where antigen is immobilized directly on the plate and detected by enzyme conjugated antibody. Here in direct ELISA, we use conjugated antibody which conjugated with enzyme to detect. Then we add there a substrate to see if there is color change or there is no color change. For indirect ELISA, the antigen is immobilized and the primary antibody bind to it. Then enzyme linked secondary antibody bind to the primary antibody. Means in the indirect ELISA, we immobilize antigen by adding primary antibody. This primary antibody is not conjugated. It is going to bind to antigen to make a complex. And after, we add enzyme linked antibody, means secondary antibody. That secondary antibody is only conjugated antibody. This antibody will added to primary antibody because it recognize antibody, it not recognize antigen. Then after we add substrate to see if there is a color change or there is no color change. Third one is sandwich ELISA, where it used two antibodies like indirect for capturing and detecting antibodies. For detecting for a target antigen, the capture antibody immobilized antigen and in the detection antibody conjugated with enzyme binding to antigen. Means direct is there to detect antigen, indirect is there to detect antibody, and sandwich is there to detect antigen, and it work like indirect, and the process are the same but different is in indirect we are detecting antibody while in sandwich we are detecting antigen. We have also competitive ELISA, which is ELISA type used to detect antigen, but also the one is coated with antigen, means antigen are going to compete to bind or no that bread or that one. Here in competitive ELISA, we use also two antibodies, primary antibody and conjugated antibody, where antibody and antigen are incubated to away to make a complex reaction, and a well is coated with antigen too. That antigen and antibody make complex, they will come to bind on coated antigen. If there is positive for patient, if there is a positive, means for positive antigen are there in that complex, or no antibody are there in on that complex, they will come and bind or no that antigen coated. But if there is no antibody there remain, it means antigen antibody they have already made complex, they will not bind to an antigen. This higher antigen concentration result in weak signal. Here there is how it is performed. Direct ELISA is addition of enzyme enzyme linked antibody, where antibody is contain is with enzyme to bind on antigen and add the substrate to get a color or no color. Indirect ELISA, we add primary antibody and we add the secondary antibody which is conjugated, and sandwich is the same as indirect, but indirect is detecting antibody while sandwich is detecting antigen. And in competitive is where we have weight coated antigen and we are looking for antigen, means they will compete to bind on, means there is a competition, is why this is called a competitive ELISA. Here I have some application of enzyme linked immunosorbent assay, where it is performed in clinical diagnostic for detecting infectious disease like HIV, hepatitis, COVID-19, autoimmune disease, and hemoglobin levels like pregnancy test. And also is applied in vaccine development, where it measures immune response by detecting antibodies against specific pathogen. Also is performed in food and environmental safety, where it detects allergens, toxin, and contaminant in food and environmental samples. This ELISA is also used in research, where it quantifies protein, cytokines, and other biomarkers in biological samples. Here I have some references where I got my information. If you are interested, you can join and may have more information. Thank you for watching my video. Thank you for your kind and attention. God bless you.